So the sign here is the 5 Hz stimulus, and orange is 1 Hz stimulus. And on the bottom, is the choice of the animal that the trials are grouped by. So you see that for 5 Hz stimuli, for trials with 5 Hz stimulus, the peak is actually at 5 Hz, very strong peak, and for 1 Hz stimuli, it's at 1 Hz. So this tells you that the intensity of the stimulus, the frequency modulation of the stimulus, is encoded in the spectrum of the response. So we analyzed all of the units like this, and on the next plot, what I plot is the difference between 5 Hz and 1 Hz normalized by the average power in the range of first 25 Hz. So this is a relative difference by how much relatively is the difference between 5 Hz power and 1 Hz power. So this is the difference for the trials on 5 Hz stimulus and for the trials on 1 Hz stimulus. And what you see here is that all the units tend to lie along this line, which says that by looking at the difference between 5 Hz and 1 Hz, you can actually tell with a quite high percent of confidence which of the two stimuli was presented, 5 Hz or 1 Hz. But you cannot say anything about the choice of the rat. So you can say about what stimulus was presented, but the choice of the rat is not known to you by looking at the spectrum. So we also did something nice. We took on each trial, okay, response of each trial, and we analyzed the spectrum of the response of each trial. And for each unit, we made a distribution of powers at 5 Hz or 1 Hz, and we looked at whether these distributions are different for stimuli that are 5 Hz modulation or 1 Hz modulation. And in the end, so this is interesting and important actually, because on each trial, the brain only has access to the information about this trial. Before here, we analyzed the spectrum of all the responses. So this is all responses for all trials coming together, but on each trial is nicer because you can say, okay, the brain could actually make a decision based on this information on this trial. And it turns out that more than half of units actually encode stimulus frequency at 5 Hz or at 1 Hz power. And some of the units, 30%, encode rat's choice. Now this is a little difficult to discuss because stimulus frequency and rat's choice are actually correlated. So it may be that what the spectrum is actually encoding is only stimulus frequency, but because of the correlation, some comes also for the rat's choice. In the end, if you imagine that the rat does the task perfectly, ideally, then this would even be the same. I mean, there would be no difference between this. So, to discuss this, we would need to analyze error trials, and for that, we would need to collect more data for the same unit, because the error trials are few. So, to summarize all the results, we trained rats to discriminate sign-modulated noise with matching velocity distributions. So behaviorally, this is a task that we've developed, and that we can now say, okay, we know how to sequence the training to achieve this kind of result. We confirmed that rats are not doing the discrimination by nonlinear integration. This is coming back to the amplitude change experiment. we looked at the spike counts and we saw that the spike counts are not informative about the modulation frequency. So this verifies the assumption that we made in the beginning about the stimuli with the same velocity distributions. And in the end, spectrum of the responses encodes identity, so modulation frequency and possibly Red's choice on a trial-by-trial basis. But to look at this, we need to analyze more trials. And this brings me to the last point. What we plan to do in the future is we plan to collect more neurophysiological recordings. In particular, we want to collect more recordings of the same unit in more sessions so that we can analyze better the error trials and look at the encoding of the Red's choice. We also plan to do information analysis on spike counts and temporal template. So this is a figure from a recent paper by Zhu et al. 2015, in which they used a novel approach developed by Panzelli and Quiroga, in which they analyzed, they were able to separate the template, so the temporal pattern, basically, of the spiking from the rate, and to quantify information contained in the spike counts and in the distribution of spikes separately, completely separately, and compare this information. And we think that this approach is very appropriate for our project. And that's all. I'd like to thank my wife, who supports me every day, and my parents, who came here from Moscow to be here with me on this day, although they may not even understand very well what I'm talking about. My supervisor, Matthew Diamond, thank you very much, Matthew. All the lab people and mechatronics lab who helped me a lot, and Francesca in particular, who helped me with training and histology. And all CISA people who have been around for all these years. Thank you. Thank you very much.
Jason, were you connected to the entire presentation?
She has the microphone.
Can you connect, Jason?
Yes, yeah. Hi.
Okay.
Hello.
Did
Disconnect you to the speakers.
the whole presentation come through on your side?
Yes.
Okay, good. So because the connection might be precarious, I would suggest that we begin with you, and then Rasmus, and then we can come back to you as necessary. Could you begin the examination period, please?
Sure, thanks. Thank you, Sergey. Did you hear me?
Yes.
Sound okay? Yeah, okay, all right. So there was a little bit of introduction, but I could perfectly follow what you were saying. Thanks for the excellent presentation. I think it's beautiful data, very carefully analyzed, nicely interpreted, nice story. Maybe I can start with a rather specific question. I wonder if I understood the behavioral paradigm properly, but you seem to have a fixed delay between the stimulus onset, the nose poke onset, and the stimulus, the start of the stimulus. I think if I understood correctly, it was 200 milliseconds. If you could go back to slide 22, where you were showing neuronal activity, I noticed there was a basal activity aligned across many trials before the stimulus onset.
Right.
For both neurons that you show. And I assume that has to do with the nose poke onset, which consistently produces that basal activity. First, I wonder if that interpretation is correct, and I ask my question.
Right. Thank you very much for the question. Yes, in fact, this activity, can you hear me well?
Yes. Yes.
Okay. This activity here is related to, most probably, to the whiskers touching the opening, the walls of the opening when it approaches the nose spoke. And as well, in the end here, on many units, you can see as well the activity related to him withdrawing from the nose spoke.
Exactly, right. So, as one way that the rat could decode this neuronal activity that we see on the screen now could potentially be to have a measure of the timing, I guess, of the burst of the activity that is induced by the entry to the nose spoke, and the relative timing at which the burst of activity which arises from the high velocity content of the first pulse produces cortical firing. And that difference, that delta T, seems to be very consistent, at least at the neuronal level, is extremely reliable across trials. Now, this is still, I believe, a form of temporal coding one could define. But do you have any insight as to what extent this latency code could be what the rat is relying on? Anything in your data that could speak to that?
So just to repeat, if I understood correctly, you mean the latency between the peak that's produced by entering the nose spoke and the peaks that are produced by the first spikes in response to the stimuli?
Correct.
Right. Well, I have not quantified specifically this value, but it seems to me that it would be somewhat unlikely because the primary reason is that in the beginning, the response of the neurons is much stronger than in the middle here. So what I'm saying is that the latency of the response to 5 Hz would be quite close to the latency of the response to the 1 Hz, just because this initial movement of the motor actually produced a lot of spikes. And even if these are, very, very low amplitude movements in case of 1 Hz, these are still sort of will produce a peak. But yes, I agree that this is something that we can look into as well, the latency between these two activity peaks.
I see. So you think the difference between the 5 Hz and 1 Hz, due to perhaps the adaptation that's produced within a packet of, let's say, the noise modulation, is not as prominent as are the number and the distribution of these patterns. I see your point, yes. All right. So maybe a little bit of a broader question. One issue with temporal coding in general is the difficulty of imagining biologically plausible ways of decoding the code from the perspective of the higher order brain areas that are reading out the activity of these neurons. So can you maybe elaborate on your thoughts on how could these sort of specific temporal patterns that you nicely show in neuronal activity with regards to the 1 Hz versus 5 Hz stimulus be decoded in a way that you could think of and that are biologically plausible?
Right. Okay. Well, we know that, I mean, it's been discussed for quite a long time, the ways that the temporal code can be transformed, in particular also into rate code. So there's been proposed neuronal mechanisms, if I remember correctly. I cannot remember the author now, but basically there's been proposed several mechanisms on the neuronal level how the oscillations in the neural activity can be transformed into spike count code. But besides that, we also have processes such as long-term potentiation and which can account for to the different responses depending on the temporal distribution of spikes. Yes, I think this is the main idea.
I'm not sure if there was a little bit of interruption in the connection, so I'm not sure if I understood perfectly your chart of correlation analysis with behavior that you showed at the very last slide. But correct me if I'm wrong, the way I understand the paradigm is that you have a noise distribution that has different seeds on every chart. So you could, in theory, every chart is in a way unique.
Yes.
And therefore that's how you get at certain properties of the stimulus. Now if you categorize, let's say, the 1 Hz charts into the ones that are correctly categorized, identified by the RAT as 1 Hz, and those that are identified as 5 Hz, so these two types of 1 Hz modulations, what are the systematic differences that you see across these two trial types? In other words, was it the frequency content of the stimulus that could tell you something about the choice of the RAT?
Right. So you're saying the difference between the trials on which 5 Hz stimulus was presented and the trials on which 1 Hz stimulus was presented? Or you mean only the correct trials?
No, I mean if we focus on one of the two stimuli, let's, for the sake of argument, just focus on the 5 Hz stimulus.
Okay.
Now you have two types of trials, the 5 Hz that are categorized correctly as 5 Hz, and those that are categorized incorrectly as 1 Hz.
Yes.
So what are the differences in the noise distribution? What features have you found that would differentiate between these two types of 5 Hz stimuli?
Right, and yes, this would be very interesting also because this potentially could disentangle RAT's choice versus the actual stimulus that was presented. And I very much wanted to look at this data. There's a slight problem with this though. The fact that the number of trials that are correct is so much higher, and the error trials are so few, because we only recorded each unit on one session. So there were no units that we can, with the degree of confidence, say that we recorded them through multiple sessions.
The question was also about the stimulus itself.
Oh, about the
To answer
stimulus
the
itself.
question, you don't even need to talk about neurons.
Oh, okay, thanks. Right, so we looked at the stimulus itself, and we did not notice any significant differences between the stimulus that was presented. That is, as you said, the stimulus is different on each trial. We could potentially find that, okay, maybe by chance, on the error trials, the stimulus has a little lower amplitude at the beginning or in the end, and we could say, okay, maybe the RAT uses this clue to discriminate. We did not see any of this kind of result. In terms of neuronal responses, the comparison is very difficult because you have so few trials. The analysis of the spectrum is really, statistically, the power is very low. So it's very difficult to draw any conclusions when you only have one session with very few error trials for each unit. That's why we want to record more sessions with the same unit.
Right, yes, I do understand, Chan. So maybe one last question before I let other people to ask questions is, I quite like very much the non-linear integration. I think that's what you call it. The idea that the 80% changes in amplitude and 120% changes in amplitude did not produce opposing effects, but rather systematic effects, in that it was as if the 120% amplitude just allows more prominent modulation, of temporal modulation, and therefore generally better performance. Whether it was 1 Hz or 5 Hz, I thought greatly supports the idea that the decoding is on the temporal patterns rather than count. And when you were answering my question, I thought also maybe this would apply to the latency question that I asked at the beginning to some extent, in the sense that even the latency between, say, the burst of activity that arises from nose port entry and the first burst of activity, that could be even viewed as a specific case of non-linear integration. So if the rat was using the latency, perhaps you wouldn't expect you wouldn't expect to see the results that you saw with the 120% and 80% amplitude. I just wanted to make sure that that's a correct interpretation. So.
Yes, I think this also applies to the latency hypothesis also. Yes.
All right. Thank you. Maybe I'll stop at that here now, and if there was time, I might get back to someone.
Thank you.
So we'll go to Rasmus, and if you could, I hope you can stay online, Asan. But I just wanted to, since one of my questions was similar to the last one from Asan, so I just wanted to quickly connect to that and then go to Asan. So to Rasmus. So it's correct that the change in the amplitude did not cause systematic biases towards one or the other, but performance did get better. Why do you think that happened?
So I think that as we increase the amplitude, the prominence in the difference in the temporal envelope comes out as stronger, and this is what helps animals to discriminate better higher amplitudes.
And could you see that in your own activity?
I did not record with the different amplitudes. This is one of the future goals as well.
So I'll start with some general remarks. So your thesis is concerns neural coding, and so your thesis addresses an issue that's absolutely fundamental in neuroscience, and the reason it's fundamental is because every time that anyone makes a measurement about the brain, they're making some assumption, whether they know it or not, about the neural code. So it's absolutely fundamental, and people have been studying it since 1926, and we still don't have a clear view of neural coding in the brain. So your approach, you set yourself a very ambitious target of training rats on this novel task, and I think anyone that's ever tried to train an animal to do something will appreciate how much work is involved in that. You've got interesting parts of your thesis where you describe things that you didn't include in your talk, that how many things you had to try before you found the version of the task that worked. So you've managed to succeed in doing a very, very, very difficult thing, and demonstrated this novel sinusoidal noisy discrimination task. So that's very, very impressive. I also, so when Matthew first told me about your work, one of the first concerns I had was about whether the rats might be solving the task using the first portion of the stimulus, where we'll come back to this in more detail. But basically, so I think it's an extremely important part of your work, this weighting function approach you took, and I very much like that aspect of your work. So, in conclusion, I think there are really there are important novel contributions to what's been a very long-running debate in neuroscience. So there's my general impression of the thesis, and I'll get to the questions. So let's pursue that point about the beginning of the stimulus. So in your presentation, you said that the stimuli, 1 Hz and 5 Hz stimuli, have the same velocity distributions, and then later on in your talk you made that more precise, because that only applies if you're considering an integer number of cycles. But I wondered whether the histograms you showed for that, are those from the kind of the numbers that come from the computer to control the actuator, or are they actual measurements of the actuator?
Right. So you're going to say this kind of right. This is a very interesting question, and one that I myself try to be very sort of thorough about is the fact that the shaker that produces vibrations has a transfer function and it affects what comes out. So we may give it this kind of stimuli, but what comes out in the end is actually, I mean, we don't quite know. So, so what we tried to do is we did offline tracking of the motor, and also we did some acute recordings that I did not include in my thesis, but we did acute recordings in anesthetized animals, and we tracked the motor at the same time, and we presented the same stimuli. So these particular histograms, they come from the input to the motor, but I checked the offline tracking of the motor with a laser finder, and I did not see the differences in distributions. So in general, there is the motor actually affects the output, but the way it affects us is in the frequency domain, and it does not come into effect in this kind of stimuli. If it was a sinusoidal vibrations, you know, with the one peak at one frequency, this would very much change the picture. But because it's noisy stimulus, and the modulation frequencies are quite low frequencies, then it does not affect, the motor does not affect the distributions.
So just pursuing that point, so that's very nice that you made measurements of the actual motor. One could still argue that perhaps the motor movements don't translate directly into movements of the whiskers. So one could, for example, make a high-speed video of the whiskers and then
one did.
One did.
Yes, well, not me, but we did in the lab. We use vibration setups for other experiments, and in other setups we did a tracking of high speed with high-speed camera. I did not do it in my experiment, but in general, the consensus is that the vibrations translate very well to the movements of the whiskers. You could track the whiskers, actually. But what's more important in this case is that the particular aspect of the stimuli that we focused on, which is the velocity distributions, should not be affected by the, say, differential translation of the stimulus to the whiskers, because the aspects that we're interested in are the same in both stimuli, which is this noisy content. So let's say if you say that, for example, frequencies after 100 Hertz are not faithfully translated to the movements of the whiskers from the movements of vibration rate, then it would affect equally 5 Hertz and 1 Hertz modulation, and therefore is sort of less relevant for the conclusions.
I mean, it's a reasonable argument, but it assumes linearity. There's no substitute, but for the actual measurements.
Well, I agree that high-speed camera tracking would be very, very nice addition to this as well.
For your behavioural data, I mean, for one of your rats, could you just give me an idea of how many trials you have? This is trials for which the animal you consider it well trained.
Right. So if I consider, for example, the amplitude change experiment. So this experiment. In this experiment, I trained 3 rats, and in the condition when they were fully trained, this is data from something like 20 sessions, and each session is about 200 trials, 200 valid trials. So say about 4,000 trials.
So linking to a point that Ehsan already made, but it might be, and this is more by way of a statement and a question, but it might be you potentially have enough data to apply some machine learning approaches to the behavioural behavior of your spine. But in principle, you could fit a weighting issue, right? You could try to fit a model to the data and attempt to actually fit a weighting function.
Right.
It might be pretty interesting.
Right. This would be very interesting, and in this direction also another person now that also works. So yes, I think we have enough data to try to combine these two research approaches and try to reconstruct the weighting function.
There was a paper by Karen Heaney that applied this general approach to
Machine learning.
yeah, also GLM model applied to some visual tasks. My next question is concerned the thesis, so it's kind of a natural break. Press ahead. Okay. So, if you go to your slide where you talk about the spike count analysis.
This one.
Yeah. So each dot here is a neuron or a cluster.
Right.
So this shows that the left figure, that at the population level, there's no difference in the cell's response to 1 Hertz and 5 Hertz. And you use that to support the basis for experiment, what we've just been talking about, the equality of the velocity distributions. But that kind of assumes that cells are homogeneous, which is not necessarily true. So there could be a completely consistent with that plot, but some of those dots stick out from the line, right?
Right.
So they could be, in principle, that you could have a subpopulation of cells that do have a statistically significant difference in their spike count response to the two
Yes. This is a very good point. I also thought about this, and in principle, you can imagine that if I sample this distribution, it can be even broader, and then the brain, we cannot say just because the neurons tend to cluster around this line that this is actually the true line, because the brain could actually only use the cells that are on the verge of the distribution, that are actually showing the difference. I think this is it's a very good point. And it is theoretically possible that some neurons would show the difference in spike count for the 5 Hertz versus 1 Hertz, and even on trial-by-trial analysis. So I also wanted to do trial-by-trial analysis. I didn't have time to complete this on the spike counts to see if any of the neurons actually support this on trial-by-trial basis. And if it's true, it would change the interpretation of this data. Okay.
I follow up on that. If in the blue plot some of the points are a little bit off the diagonal, some of the cells or clusters are a little bit off the diagonal, if those were important, if those distant differences in response to two stimuli were important in the decision making, would you predict some connection between the left plot and the right plot?
Right. So if this information in the spike count is actually used to guide behavior, then we would also see more prominent that the spike counts predict the choice of the animal, along with also the stimulus.
And would you predict that an individual neuron or neuronal cluster, if the difference from 1 Hertz to 5 Hertz were important in decision making, then that individual neuron would also show enhanced difference in the left versus right?
Yes. Yes, certainly. This is the expectation that I would have. If this information is useful. I think when we do information analysis as well, we will extract this data and we will see the differences for different units. If some units use this information if they encode the choice better, then we will see.
I think this cell-by-cell analysis is important because, as far as I know, no study on behaving animals in cortex has ever found all the cells to be involved in whatever decision is being dealt with. Always, the results are really concerned with a subset of significant cells.
Yes, I agree.
Just going back to the RAS case, a question that's almost the same as a question that Ehsan asked. So, Ehsan asked about the burst of activities, the pre-stimulus. Right, and after the burst and before the stimulus comes a dip.
Right.
And same a dip at the end of the stimulus as well.
Right, so this dip actually comes from the pre-stimulus delay. So if I go back to the sequence of the events, before the stimulus and after the stimulus, there is a delay that's about 200 milliseconds. Actually, in some sessions I varied it as well, but in most sessions it was fixed.